Hi everyone. Today I'm going to walk you through how oxygen binds to Heme inside hemoglobin, the structural changes that will happen when it binds, and how that changes oxygen delivery in different parts of the body. We will look and see what happens in the lungs compared to the capillaries, and we will also talk about how carbon dioxide interacts with water, lowering the pH of blood, and why that makes hemoglobin release oxygen more easily. Altogether, this shows how our bodies are really fine-tuned for oxygen transport. Heme is a small but crucial molecule inside hemoglobin that directly binds oxygen. It's made of a porphyrin ring, which is a flat, ring-shaped structure, and its center is an iron ion in the +2 or ferrous state. That iron can make 6 bonds total. 4 of them are locked into place with nitrogen atoms of the porphyrin ring. 1 bond connects to a histidine amino acid from the globin protein, and the 6th spot is open, and that's where oxygen binds. What's so important is that oxygen binding is reversible. That means oxygen doesn't get stuck forever, and hemoglobin can act as a delivery system, loading oxygen when levels are high, like in the lungs, and releasing it when levels are low, like in our tissues. This reversible binding is what makes Heme and hemoglobin the perfect oxygen carriers. When oxygen actually binds to the iron, it pulls the iron atom slightly down into the plane of the porphyrin ring. That tiny shift creates a domino effect that changes the entire shape of the hemoglobin protein. Hemoglobin exists in 2 main states: the tense state, which stands for tense, and the R state, which stands for relaxed. The T state doesn't like to bind to oxygen, and the R state binds to oxygen much more easily. When the first oxygen binds, hemoglobin starts switching from the T state to the R state, and it makes it easier for the next oxygen molecule to bind. This process is called cooperative binding. It's why the oxygen dissociation curve is S-shaped or sigmoidal, and then on the next slide we'll take a look at that dissociation curve instead of a straight line, which oxygen molecules that binds make it easier for the next to hop on until hemoglobin is fully saturated. Without this property, hemoglobin would not be nearly as efficient at loading oxygen in the lungs. So you can see here the changing of the forms that I stated earlier. So the lungs versus the capillaries. Now let's compare what happens between them. In the lungs, oxygen levels are very high. Because of that, hemoglobin is almost fully saturated, close to 100%. That means nearly every hemoglobin molecule is carrying its maximum load of 4 oxygen molecules. The pH in the lungs is about 7.4, which also helps hemoglobin hold on to oxygen tightly. In contrast, in the tissues and the capillaries, oxygen levels are much lower because the cells are constantly using oxygen for metabolism. When hemoglobin enters these lower oxygen areas, it starts to release its oxygen. This is exactly what the body needs: oxygen delivery to places where it is being consumed. The sigmoidal dissociation curve, which is right here, you can see it, shows this beautifully. At high oxygen levels in the lungs, hemoglobin loads up. At lower levels in the oxygen levels in the tissue, it unloads. This contrast between the lungs and the capillaries is essential for our survival. The Boer effect. In the capillaries, another factor comes into play: carbon dioxide. As our cells work, they produce CO2. That CO2 diffuses into the blood and reacts with water, forming a carbonic acid. Carbonic acid quickly breaks apart hydrogen ions and bicarbonate. These hydrogen ions make the blood more acidic, lowering the pH. When pH drops, hemoglobin's affinity for oxygen decreases. In other words, hemoglobin doesn't hold on to oxygen as tightly. This makes it much more willing to let go of an oxygen in the tissues. The effect where lower pH and higher CO2 causes hemoglobin to release more oxygen is called the Boer effect. And you can see here, they're releasing in terms of pH. The Boer effect is another layer of regulation that makes the oxygen delivery system really smart. In areas with high carbon dioxide, meaning that the tissues are working hard and burning a lot of fuel, hemoglobin automatically delivers more oxygen. It's an elegant feedback mechanism. So, in summary, to sum up everything, oxygen binding to heme pulls the iron into the plane of the ring and shifts hemoglobin from the tense state to the relaxed state. That conformational change makes hemoglobin cooperative, so once oxygen starts binding, the rest bind more easily. This explains the S-shaped dissociation curve. In the lungs, where oxygen is abundant and pH is stable, hemoglobin loads up with oxygen. In the capillaries, where oxygen is low and carbon dioxide is high, hemoglobin releases oxygen. And the Boer effect fine-tunes this process by ensuring that tissues producing the most CO2 get oxygen released. Altogether, these mechanisms show how hemoglobin is perfectly adapted to balance oxygen pickup and delivery in the body. All right, well, that's it. Thank you, guys.